Okay. Hi, everyone. Thanks for being here. I'm excited to introduce Dr. Ben Hayden today. Although I always think of Dr. Hayden's work as revolving around naturalist and economic behaviors, when I was looking up his CV, I actually realized that he did his PhD with Dr. Jack Lant, which I hadn't realized, where he worked on the role of attention and visual processing. He then moved to Dr. Michael Platt's lab for his postdoc, where he worked on the role of ACC, or the anterior cingulate cortex, in foraging and reward behaviors. And since then, Dr. Hayden's lab has continued to pioneer how to study the nervous system during naturalistic behaviors. And I think has been really pioneering new quantitative and experimental methods to study them. So I'm just going to give you that's going to be the end of the brief introduction because we started a bit late. Ben, thank you for being here, and I'll leave it to you.
All right, thank you, Adam, and thank you for the invitation. I appreciate that. I hope everybody can see this little monkey here out on the field site of Cayo Santiago in Puerto Rico, where I've done a little bit of field work over the years. Let me also just thank the organizers for inviting me to this series. It's a bunch of really amazing people talking, and I'm really honored to be among these group of people talking. And I'll also just say one more thing, which is, I already said this to Adam, but now I'll say it to everybody. If you have questions, ask them. I think you're going to have to communicate them to Adam. I can't see anything that's happening other than what's on my keynote screen, but I told Adam to just interrupt if it seems like it's an urgent question. I also put my email address on this slide here because I want you to err on the side of not caution, of just sending me an email if you have any questions about anything you see or anything else. The title of my talk is The Neuroscience of Naturalistic Decisions, and that's kind of the focus of my lab. And so my background in neuroscience, after I did vision and attention for 5 years, has been really in the world of neuroeconomics, of making decisions and choosing, and using rhesus monkeys as a model just so we can get brain activity. And the goal of that research has really been to understand how we make decisions, but with a focus always on naturalistic decisions. So, I put this here to remind myself to mention, this is what I've actually been spending the last 3 months doing, is dealing with my baby. And so, if I'm a little bit rusty, that's what's going to happen. I'm going to be thinking about that baby. But also just to say, I really like this baby, and I'm very proud of the time that I spent with her and the benefits of the pandemic. There's been costs, of course, too, but it's been nice to get a lot of time with her. So lab studies, economic choices, and just to repeat myself, we usually in neuroeconomics think of choice as being binary decisions between two discrete options made at a discrete point in time. There's one unambiguous reward that you get, and that's been a very, very productive approach. That's been the bulk of my approach, but we've always been pushing it and nudging it towards the direction of making it more naturalistic. And the ideal, of course, is to understand, if you're interested in monkeys, is to understand how they make decisions in the naturalistic context, in a world in which they are both embedded in that world and they're embodied within their decisions. And there's a philosophy behind it, and the philosophy is, these are the ways, this is the types of decisions the brain was evolved to make, and there are going to be things that you're going to miss if you don't take naturalness seriously. And I'm not going to spend a lot of time, because I assume the fact that you're coming to this talk series, you're probably already at least halfway convinced of this, so I'm not going to spend any more time on that. I'll just give you this quote that I saw on Twitter not too long ago. Ethology is interviewing animals in their own language, and this is, of course, a famous quote from Tinbergen, who's shown here. I'm going to kind of skip this or not go into much detail on this and say there's a lot of benefits to taking the naturalistic approach. You get qualitatively new types of behavior, new types of neural activity. And I'll also say, because Adam mentioned it, although my background is in vision, when I was in Jack's lab from 2000 to 2005, the big focus, the big centerpiece of all that research was natural vision, with the idea that if we move beyond gratings and bars towards naturalistic stimuli, we're going to have a much better ability to characterize visual responses. And I actually think of the stuff that we're doing here with decision-making as really as being intellectually continuing of that, with the idea that natural is different. Natural does put your brain into different modes, and those modes are more realistic and more valuable for understanding the brain. So we're always going to be pushing, and I'm going to talk about two different stories today. I had to be very ruthless just so I could give a little bit of detail on these stories. I'm not going to talk about everything that's happening in my lab. This is just a thin slice of it. But by doing that, I hope to give each one enough time and that you can actually sort of understand what we're doing and why we're doing it and how we got to the point where we want to do that. So I'm going to start by talking about what we call the avatar-based pursuit task, but of course we actually call it the Pac-Man task. And this is a project that was really developed by a really smart postdoc in my lab named Alex Tomei several years ago. He moved on to a government job, but the banner was picked up by this guy, Michael Yu, who just started a faculty position straight out of graduate school in Korea. And he got permission from the government of Korea to do a brief postdoc with Mehrdad Jazieri at MIT. But he's gonna continue this work, and so we're gonna work on this together moving forward. So here's the actual task. I like to show this video. Can you guys see this video? Is it moving? Sometimes it's not moving when I do this on Zoom type things. Adam?
Yeah, it's moving.
Okay, good. Sometimes it's frozen. It's moved. Sometimes it moves for me, but it's frozen for everyone else. I actually see pretty good here. So one thing I like about the task, this is called the pursuit task or the Pac-Man task. As the task gets more complicated, I think if it's designed well, and we like to think this is designed well, you need less explanation for how the task works. I hope you can see there's a monkey on the left here, and he's controlling this joystick. Once the monkey learns how to use the joystick, it's very quick, only a couple days, certainly less than a week, to train him to do this task. And then for about 2 months, the monkey will get better and better and better, and then he'll kind of plateau. And so that's when we were going to record here. The rule of the task is very simple. The monkey's joystick corresponds to the position of this yellow circle, and he's chasing these delicious, yummy colored squares. And if he ever captures the square, he gets a squirt of juice. If he doesn't capture the square within 20 seconds, then he doesn't get a juice. That seldom happens. And then on some trials, there's these nasty, scary triangles that pursue the monkey. And, he really hates those, and he tries to avoid them. But, of course, by avoiding them too much, you're going to impair your ability to capture the square. So there's a bunch of really great decisions the monkey has to make here. And the more we think about this task and the more we work on this task, the more we think this is really a near ideal task to study a lot of questions that are sufficiently controlled that, we're not out in the wild, we're not out in the Cayo Santiago field site doing everything, but it's significantly more complicated than the types of tasks that our lab, at least, has been studying for the last several years. And we're getting more and more interested in this task. So in our mind, in my mind, in Michael's mind, as well as our collaborator, John Pearson, this task is a near ideal tool for studying continuous decisions. And so we distinguish between discrete decisions, which like 99% of neuroeconomics, including our own work, is focused on, and continuous decisions. And if you think about it, while you're making this decision, while you're the monkey moving this joystick around, you have to move the joystick. You can move it any one of 360 degrees at any moment, and you can change it continuously, and you can alter it, and you can adjust it. And the options you have at any moment depend entirely on the choices that you made the previous moment. The time is really continuous, action is really continuous, and you don't really get rewarded except very occasionally relative to the number of decisions you make. And so while the neuroeconomic approach of, calculate the expected value of each possible option is in theory going to be the right approach, it's actually really cumbersome to kind of solve the Bellman equation and do all this stuff. In practice, we think that starting with control theory, engineering control theory as a basis, using objective functions instead of value, is going to be much more useful way to start solving these problems for us as neuroscientists trying to understand the behavior of monkeys. And again, this task, we don't think of it as particularly weird or unusual. It has a lot in common with the bug hunting behavior of monkeys, where, there's a bug flying around in front of you and a monkey will reach out and grab it, either to swat it or to eat it. Humans do this very readily. Monkeys seem to enjoy this task. So to humans, it seems to be very intrinsically rewarding. So that suggests to us that we're not doing something weird and unusual and training the monkey into something that his brain is not exposed to, but rather something that taps into some very standard, very evolved mechanisms. But again, I'll acknowledge this is not purely naturalistic. He's using a joystick. It's several levels removed from nature. These are little shapes on a screen. So we're trying to compromise between tractability and naturalness. And so, I think that's going to be a theme for all of probably this whole talk series, is that compromise. But we like this because it's one step beyond what we've kind of done in the past with the apple and the orange slide that I showed at the beginning of the talk. All right, so I won't bore you with too many details because I already showed you the important part. There's a bunch of different prey, that's a bunch of different squares. They're indicated by colors. They're different velocities. So they're harder to get, and there's a bunch of different predators, and they have different costs as well. Just to kind of illustrate some points that these monkeys don't really go into stereotype behaviors. These tasks evoke a very rich repertoire of behavior. The monkeys' behavior is not purely equally spread out across the screen. They have certain places they spend more time, and those are determined, of course, by the physics of the task. But they do tend to capture the prey at a relatively broad spectrum of places, and they do tend to travel pretty broadly in the screen as well. And of course, we're going to record brain activity while we do this, because that's our interest. We want to understand ultimately how the monkey makes the decisions that he makes in this task. Basically, we want to treat the brain as a black box that generates behavior given stimuli, and we want to understand what its transfer function is. So that part of this task is not too different than the way that we've always done neuroeconomics. We want to understand how the brain transforms options into choices, but now the mapping function is going to be a lot more difficult because the task is a lot more difficult. We're interested in all the parts of the brain, of course, but I'm going to just talk about the one brain area we've actually recorded in this task, in the dorsal anterior cingulate cortex. And if you don't know much about that, the way I would describe it, the way I like to think of it, is as a premotor cortex. And so it kind of sets the stage for premotor cortex. It kind of sets the stage for the guy that sets the stage for motor action. It has a very high level, very executive role in controlling behavior. Gets very strong emotional inputs, cognitive inputs, and of course it does get motor inputs, although we think of those as feedback type inputs. And I'll just say we call this area 24 in the lab. Other people, there's a lot of dispute in the literature about what number to give this, and I'm not going to go into that today, although it's a fascinating question. All right, so the main thing I want to talk about today, just for this task, before we move on to the other stuff, the other half of my talk, is that when you're doing a continuous decision-making task, when you have a very rich and complicated environment that's much more complicated than just apple versus orange, you have this extra problem. You have this qualitatively new type of problem that we don't really even have to think about or deal with, which is we have to map the world. We have to think about what the structure of the world is, and we have to be able to use that structure to drive our decisions. That's really the foundation of our decision-making. And again, in a two-option choice test, you have to represent the values of the options and compare those, but that's a trivial question. It's a much lower dimensionality question than the mapping problem that you have. And that's going to directly drive actions. And so our fundamental hypothesis that we've now published 2 papers dealing with is that keeping track of your spatial position is something that's so rich and so robust, it's probably found repeated in many parts of the brain, and certainly it's going to be found in the dorsal anterior cingulate cortex, this premotor structure.
Ben, there's a quick question. I don't know if I'm raising my hand down here or what for you to call me. But Rampant Soup asks whether there's some uncertainty in the task once the target and pursuer are on the screen, and also whether some pursuers are more likely to catch the monkey. They said they saw the pursuer speed, but not. I saw the yeah.
Okay, so there's 2 questions there. Yeah, so those are good questions. The pursuer is kind of dumb. It's just doing an A-star algorithm, which I think is the same algorithm that the ghosts in Pac-Man used. It's a very old classic video game algorithm for pursuit, which is basically just going to where the monkey is. The AI is not different. It's just the maximum speed that those different predators can have. That's the only difference. There's a whole bunch of really interesting like theory of mind and strategy and stuff that you can pursue with this task. We haven't done that yet, but those are directions we're very interested in going. The other question, what was the other question? The other half of the question?
Whether some pursuers are more likely to catch the monkey. They saw that like pursuer speed was a parameter, but not probability of capture.
No, there's no difference between them other than speed, but the faster ones are definitely more likely to capture the monkey. And one thing I can tell you is that we see the monkey is actually willing to be pretty tolerant of the risk of getting captured. The cost is not too high. It's only about 5 to 10 seconds. And the monkey is definitely much more willing to let the slower predators get closer to it than the faster predators. So the monkey does seem to recognize that there are different levels of danger, but that danger comes only from the dimension of velocity. Should we stop and take any other questions? Let me just see if there's any other questions. Anybody else want to
jump? Does anyone else have any questions? You can add it to the chat window. I guess the downside is it takes a little time to add something to the chat window.
Yeah, well, I'll just keep going then while we're waiting and see if anything pops up. So as I said, kind of the foundation for understanding the role of these neurons and the role of this brain region in naturalistic or complex decision-making, including in this type of task, is to understand the mapping function, how it represents the structure of the world, right? So the more interesting questions we're going to get to in future years, we haven't even been able to tackle yet because we need to understand the first problem of mapping. You're probably familiar with place cells and grid cells, which are associated with hippocampus and medial-entorhinal cortex, respectively. I want to just talk about briefly this very exciting paper that Kia Hardcastle published when she was in Lisa Giacomo's lab and worked with Surya Ganguly, where they looked at medial-entorhinal cortex in rats, and they found that while you can find this beautiful grid-like patterns, there's a whole bunch of non-grid-like cells in the same brain region that also have just as much information about space as the grid cells. They're not nearly as famous because they're kind of blobby shaped. And they're a little bit, the math behind them is a little bit harder to develop, although they developed it in this paper and we copied their math. And so we took their approach largely because we didn't want to go in a priori with the assumption that neurons in ACC map the world using a place or a grid cell-like structure. We wanted to let them do any kind of structure and just see if there's any mapping at all. And that's what we did. So we see some data here that is in a paper that actually was recently accepted. It's on bioRxiv now, and it will be published pretty soon. What you see in these little rectangles on the right-hand side of the screen in panel B is a heat map. The X and Y of the heat map correspond to the screen. And so left on the little heat map corresponds to the left side of the screen and so on. The Z-axis, the color, indicates the strength of firing of a single neuron. So these are just very particular cells that we've selected from a very large pool that we collected. And you can see that these neurons have aperiodic, non-place-like, non-grid-like tuning functions, but they do carry information. And this is not just statistical noise, as was, this is the real advance of the method that Hardcastle and colleagues developed, is you're able to find real, non-periodic, non-grid-like structure in tuning functions. And so these are just some example cells, and I'll just walk you through. Well, let me just say simply that these three cells are very typical in the sense that they show selectivity for the positions of the self. That is, when the monkey's avatar, when Pac-Man goes into a certain part of the screen, the neuron's going to fire, and when the avatar goes into other parts of the screen, the neuron's not going to fire. So they carry, and so collectively, the population carries a very rich map of where the self is. They also carry information about the prey and of the predator on the trials when there's a predator. And they're different maps. So a cell, like if you look at session 5, cell 16, is going to be especially likely to fire when the self is on the right and also when the prey is on the left. And these maps look like they sum linearly. So there's a very rich map of space here in DACC. And this is something, I mentioned at the beginning of the talk, that by doing something more naturalistic, you're going to be able to see patterns, see things that you wouldn't be able to see otherwise. You wouldn't ever see this in a simple economic choice task because you don't have that dimensionality. You don't have the high dimensionality spatial map. You just wouldn't even be able to ask this question with your data. So we see this very rich repertoire of information that is found in this part of the brain, not normally associated with mapping, more associated with executive control, but it's in there and it's real, and you can detect it with these novel statistical methods. There's also other stuff. I'm not going to go into this because I'm already taking too long, but I'll just say variables related to the relationship between the self and the prey are encoded in these DACC neurons. So what these heat maps show is if the monkeys, whichever direction the monkey's Pac-Man is going is by convention up on this graph. If you just look at the first heat map here, session 1, cell 15, when the monkey's going up, if there's a prey that he wants on his right, you're going to have high firing in this cell. And if he's a prey that he wants on his left, you're not going to get any firing. And so these are second order or egocentric variables that are also found within the DACC and the brain of the monkey.
And there's two, I think, good questions about the previous result. The first is whether the monkey subjects need to keep fixation in some way.
Fixation is completely unconstrained.
Okay.
But we control for fixation. You don't see a lot of fixational tuning. You can regress that out pretty easily. There is some, but it's much weaker than the I would say the behaviorally relevant spatial variables.
And the second is whether it's an active thing. So if you played a video of a previous trial, would you also get these maps?
We never tested that. My hunch is that it would depend on what the monkey is thinking about when he's watching the video. If he's like mentally imagining himself doing that, which is what I do when I watch some people play video games, you probably would get something, would be my hypothesis, but we've never tested that. That's a good question.
And do the monkeys follow the prey or the predator, or do they look at their own position in between?
Great question. They basically go back and forth between the self and the prey, and they don't spend much time looking at the predator. It looks like they tend to do that through their peripheral vision. They basically tend to ignore the predator until it gets pretty close to them. But again, we haven't gotten that much into the details on this. One thing I like about these questions is they reveal this is like a very, very rich data set, and there's a whole bunch of things you could ask. And we've already written 2 papers about this. We're writing a couple more, and none of those questions are in any of the papers because it's such a rich data set. And so we're kind of like overwhelmed with the number of questions that we could ask. But these are all good questions, and if there's any more, I'm happy to take them too.
That's it for now, but I'm sure people will come up with more.
I'll just interrupt myself and say you're probably wondering at this point, I told you about egocentric and allocentric cells. I didn't use the word allocentric, but that's what the first cells I talked about. Are these different cells? Are the self, prey, and predator cells different cells? This ties into a different topic that my lab has spent a lot of effort on, which is detecting the functional classes of cells. And this has been like 4 or 5 papers. The culmination is this Jane Neurophys paper that I wrote with Tommy Blanchard, my first graduate student. This is his last paper. We are pretty certain that we can say with high degree of confidence that these are not only are these not different cells, but there do not exist discrete functional classes that you can detect a priori using functional properties in this brain region or in any other prefrontal region. So largely these properties are highly mixed. You get mixed selectivity that seems to be, this is a kind of a big debate going on right now with Stefano Fuzzi being the big proponent of mixed selectivity and other people disagreeing with that. Basically all our work, including I think some very advanced statistical methods, say that Stefano's approach is completely correct and that a lot of the theorizing that he's done about mixed selectivity seems to be valid. Simply put, there basically aren't egocentric and allocentric cells. All cells that are task active are both. All right, so I'm going to say one other thing, and then I'm going to move on to the freely moving monkeys. And that one other thing is going to take about a few minutes. So in naturalistic tasks, in addition to mapping the world, you have to constantly predict the future. You have to guess what's going to happen. And we've done a little bit of work on this problem of, how presentation of rewards leads to representations of the potential future from those rewards, mostly in Maya Wong's paper in Nature Communications. This is using a very conventional task. In the Pac-Man task, predicting the future is much more complicated, right? If I show you a little orange bar or blue bar, and the blue bar is associated with 250 microliters, that's like a very low dimensionality type of prediction. But in the Pac-Man task, you basically have to say, well, if I move this way or slightly more that way, that's going to create an entirely different world with an entirely different set of opportunities and costs. It's like mind-bogglingly complex. And that's kind of why we're so fascinated about it. We're going to ask the very, very simple question, though, to start with, by saying, does the monkey actively predict where the prey item? Let's not even talk about the predator. Let's just think about the prey item, which is the simpler case. Does he think about, and there's just imagine the one prey case. Does he think about where that's going to be and head towards the position where it's going to be in the future? Or does he do the much simpler thing of just pointing the joystick towards where the prey is now? Or does he even do the even dumber thing of just seeing where the prey is now, and then with some slow reaction time, pointing to where it is right now, but he's going to lag the prey? So functionally, does the monkey lead the prey, point at the prey, or lag the prey? Okay, and you can sort of see the logic. And we did a complicated analysis. I'm not going to go into detail. It's in this paper, and this is kind of the heart of this paper is asking this question. And the answer is the monkeys lead the prey. Monkeys are basically doing this red arrow here, solid red arrow. They predict where the prey is going to be, and they are more accurate than chance. They do some basic physics. They take we can show that they take account of the velocity and the acceleration of the prey, as well as its position, obviously. And that makes their prediction more accurate. And in fact, across 3 monkeys that we ran this on, they tend to predict the position of the prey somewhere between 700 and 800 milliseconds in the future, which is pretty impressive, given that the whole task typically takes about 5 seconds per trial, 5 to 6. I'll also say that when you start the monkey on this task, they tend to lag the prey. They tend to go behind it. After about a month, they get to the point where they can predict the position of the prey, and then they keep doing that from now on. So they get better and better with time, and that improvement tends to look roughly linear. So there's an active prediction of the future, and the monkey heads toward that position that improves their performance by about 10%. They get about 10 to 15% more juice each day by doing this future prediction. And this is something that's very, very interesting to us, because predicting the future is probably one of the things that the brain is designed to do. It's probably one of the reasons we have brains and don't just have direct connections from our eyeballs to our hands. Let's see, I'm going to skip this because we're running out of time. I'll just say there's all these kinematic variables, and we find evidence for coding of these variables. All right, well, here's the interesting thing. We also find an explicit map of the future prediction of the prey in the brain, and this is distinct from the map of the current position of the prey. So if you look at these yellow squares over here on the left side of panel A, you can see that this neuron, this one particular neuron, tends to fire when the prey is over in the upper right-hand quadrant of the screen. And it also tends to fire when that prey is going to be, when the monkey accurately predicts that it's going to be in the lower right-hand quadrant of the screen. And of course, these maps, which again use the algorithm that Hardcastle and colleagues developed, are not as simple as like quadrant versus non-quadrant. They're much more complicated. But by and large, you see very little similarity, at least in this neuron, between the map of future position and the map of current position. And overall, they are separate maps. Although there are certainly some cells, if you look at this top 2 cells in the top row here, that showed the same, basically the same map. So we see a whole range of similarities. The inset line graph here just shows you the range of similarities. You see highly dissimilar like the yellow and highly similar like the purple. Okay, so the point is you have a rich map of the explicit map of the predicted future position of the prey as distinct from the current position of the prey. So, all right, I'm going to say one other thing, which is the future directions, and then we're going to move on to monkeys moving around the cage. But before we do that, we'll take any more questions. I'll just say the next step, the future directions for this task, is looking at decision-making in this task. And so decision-making in this task is not as simple as maximizing the expected value of an option. It's actually much more complicated. We basically have to build a model, a generative model that predicts the monkey's behavior, and we're going to have to show that that model is highly valid predictor of the monkey's behavior, and then we can start querying that model. And so we build this model. We're at the point where we've built the model. It works pretty well, and we have not yet started using it. But basically, it's a virtual monkey. It's a virtual Pac-Man player that we can feed it, a problem. We can feed it a starting position and velocity and acceleration of self, prey, and predator, and have it try to guess where the monkey's going to go. And it's going to be stochastic, but we can still ask the question of how close it is, how accurate it is. And so we're mostly done with this. We're not totally done yet. Once we're done with that, we're going to start relating this to brain activity and start seeing how this is implemented in the ACC as well as in other brain areas that we haven't recorded from yet. So, oh yeah, and so basically, this model, what it gives us is a cloud of goal states. It tells us what the goal state of the monkey is, and we're going to start by asking how that is represented in the brain. All right, so I'm going to pause right here. I think I'm about halfway done, maybe a little bit. We started a little bit late, but I'm going to talk about some monkeys moving around in the wild, but I want to stop and answer any questions about this Pac-Man task before we go on.
So the only one that's come up so far, and if anyone else has any questions, please put them in the chat or in the Q&A section.
And also, I left my email address at the beginning. You can find it by Googling it. You could just email me if you think of it the next day and you're like, oh, I wanted to ask that question. Just send me an email.
Lauren Strickland asked whether the 700 to 800 millisecond prediction is similar to humans.
No. Well, that's a great question. I will tell you this. If you watch these monkeys, I can't watch the monkeys do this task because it gives me a headache. They are so good at it. But they've been doing it for 4 hours a day for a couple months when we start recording. They've been plateaued for a month. And then they continue to get slightly better throughout that time. They get so good that it hurts your brain to watch. The video that I showed is from like 2 weeks of training. They get so much better that we kind of have to keep ratcheting up the task and making it harder. Humans have never done that. We've had people do this task, but after like 2 hours, we're like, okay, here's your $10, go home. We haven't had people do it for like 2 months straight. If we did, I suspect that humans would be the same as the monkey, but comparing human to monkey performance is something we're very interested in. I think, we would have to have people basically play it the way that I played video games when I was 10 years old, like obsessively, for months at a time. And if you watch me playing Sonic the Hedgehog, you would also get a headache doing that too. But right now, I think my intuition is humans and monkeys are basically no different in this task.
And Monica Rodriguez asks whether you're able to look at variables such as sex and age, and whether there's a specific age you start them on the test.
Oh, that's a great question. Yeah, you need a lot more subjects. We've only ever run 6 monkeys on this task. There's not enough to do any statistics on. And they're all males, and they're all, they all start when they're 3 or 4. Those are great questions, though. We'd love to do it. It takes a lot of time to train a monkey. It's a very bespoke type of experiment. So we haven't been able to do that, but it's a good question.
And then Rampant Soup asks again, they think they had an intentional lapse. Did you say that most of the selectivity is for task-relevant features, but not specifically for egocentric and allocentric positions? And do you see maps for relative distance and position instead of fixed?
When I assume relative means self relative to prey. If the answer is yes, most selectivity is not particularly for. I mean, here's kind of the data on that. The strongest selectivities are the positions of the self, prey, and predator, and then you see derivative of self and weakly, but significantly, positions of the prey. The predator we don't see. I would guess that if we had more data, we might see that crossing over the significant line, but it doesn't at this point.
Mm-hmm. Okay.
So yeah, so basically position is the strongest encoded variable, but they're all in there somewhere. As you can see from from panel E, there's a lot of neurons that encode multiple variables simultaneously.
Okay, that's everything for now.
All right. Well, that's the hard part. Now I'll just show you some fun videos for the next 20 minutes. This is a little gift that I made. All right, so I'm going to tell you about the freely moving monkey task, which is kind of the Pac-Man task squared. And so kind of one of the big dreams that I've had for 20 years, and why I went to Michael Platt's lab 15 years ago, was to start understanding the physiology, the decision-making of monkeys doing something even more naturalistic than Pac-Man. This has been an incredible amount of work. I basically started this the day I got tenure in 2016, 5 years ago. And I roped in Jan Zimmerman and Hyun-Soo Park to help me with this. I definitely could not have done it without them. And in fact, it's basically an equal share between the 3 labs at this point. And we have a bunch of people, including some other people who've already moved on. This is how long this has been going on. But we finally kind of cracked this nut, I think. And so this is the paper that just came out last year that put aside the physiology, which is actually a much simpler problem. Just tracking where a monkey is moving around in space is an incredibly difficult problem, and it took this whole team of people plus a bunch of other people to kind of get there. What we did is we built this cage in our lab at the University of Minnesota. It's basically 9-foot cube, and I guess I should say 3-meter cube because we're using the metric system. And it's surrounded by, it's a clear cage. It's mesh cage, 1-inch mesh. It's surrounded by 62 machine vision cameras, and just setting this up and getting these computers to talk to these cameras to talk to 6 enormous servers so we can get them all simultaneously, not drop any frames and stuff like that. It's a real headache. We generate over 3 terabytes per hour of data. And then you get the monkey in here, and there's a whole world of stuff I could talk about for days about just training the monkey to do these tasks. But basically, there's little interactive feeding stations that the monkeys have dispersed, and we can move them wherever we want. And they like doing this. They love being in this cage. The university loves it because they find it enriching. They believe it's enriching. We do too. We painted it green because of the same reason that Hollywood uses a green screen. It's just easier to segment the monkey against the background when you paint everything green. We literally rented a spray painter from Home Depot and a bunch of green paint and painted it. And this is actually chroma key green, the same color they use for green screens. Here's another view of it. This is actually before we painted the barrels, which we have now done also to improve segmentation. Here's just an example of what you get. This is 16 of the cameras. We get coverage of pretty much the entire room, pretty much every square inch of it. We get several cameras looking at it. And so the goal here as a math problem is if you just take all these pixels, try to decompose and figure out where the major joints of the monkey are. And that problem, beside the technical problem of building this and training the monkey to do it, is the major focus of the last 5 years of the lab's research. All right, here's the cage. All right, so Open Monkey Studio. I will say, because everybody asked me that at this point, the heart of my Open Monkey Studio is CPM, Convolutional Pose Machine, which is a very standard off-the-shelf thing. It is not very different from Deep Lab Cut. They are quite similar, and in fact, you could. We haven't done this, but you could replace CPM with DLC, and you'd get the same results. The engine is not the hard part of doing this, and so it's just getting the quantity of data and a bunch of other things that are going to allow you to boost, given the number of images we have, the effective number of images. The bottleneck is annotation. So when you track monkeys, you can think about this as really hard problem that has been solved for humans. And the way it was solved is that the Silicon Valley put about $12 million into annotating an enormous number of images, and that those annotations are really the core of how human tracking works. We don't have $12 million. You need that to do the equivalent job with monkeys, and we want to do the equivalent job. So we're going to use a bunch of tricks that are developed by Hyun-Soo Park, our computer science collaborator, and we're going to use a large number of annotated images that we get, and we're going to use a bunch of things that you can do because we have 62 cameras, where we have cameras 1 and 3 tell this, and we can guess what's happening on camera 2, which is in the middle of them, and check and see if that's right, and basically augment our ability to do the tracking, stuff like that. All right, just in case you're wondering, why are macaques are actually harder than humans, and they're definitely a lot harder than rodents. They're basically giant balls of fur. They're very hairy, and we've definitely spent many an afternoon debating whether we should shave our monkeys, but we don't want to do that for a lot of reasons. One is their hair grows really fast. They move extremely rapidly, much more rapidly than rodents. They have many more degrees of freedom. They move in 3 dimensions. There are many more rare poses, which actually turns out to be the major difficulty of these types of things, which is also why humans are hard. So these are creatures that will often walk around on the ceiling. And you can see in this picture here, this guy is just doing this effortlessly to try to get some grapes that we threw up there for him. We used a marker-based system called OptiTrack, and we're able to show that our system, our computer-based system, is actually more accurate than OptiTrack is, because OptiTrack has a bunch of dropout that you have to average over and smooth over, and we don't do that. So the result of this, the result of all our work, is that we can track the position of the monkey. We have 13 joints. We could get more, but it would take a lot of effort to do. So one of the big things that we're working on right now in the lab is improving this. So Praneet has a project that with a very, very small number of annotations, so just 568 annotations, he's able to add new joints, and he's basically able to have a deep net that is so intelligent and has such a good internal understanding, a representation, I might say, if I were reading Twitter a lot, of the physics of how the monkey's body moves, that if you give it only 568 examples of the elbow or the tail or the middle of the back or some other joint that we hadn't given the annotation process, that it's able to generalize and do a very good job, a very high degree of accuracy of generalizing. Hao Zhang is working on a project where he's basically draping the monkey in a mesh in software and trying to track the position of the entire body, which is basically a 40,000 point annotation as well. And so we're going to basically be able to get a very high degree of tracking from this system. And we can start asking, even more sophisticated questions. We're also, including like where the 5 different digits of the hand are in this environment. We have the capability on the camera. We just need to do that with our tracking. I will also say, because everybody asks me at this point when I talk about it, what if you are doing field research like this? This is me 10 years ago when I was younger and had shorter hair, non-pandemic hair. What if you have one camera, which you often have in field research? So the idea is the model that we generate from the 62 camera data has within it a lot of information about the way that monkeys' bodies move. And so if it's good enough, and we're not there yet, but we're close, you should be able to apply that model to a single camera and do some guesswork about where the monkey is going to be. And in fact, you can even, with even a rudimentary model of the environment, you can lift, which is guess the depth in the 3rd dimension of where the different parts of the monkey are, which is crucial for doing good motor neuroscience. And so this is a software product called Open Monkey Wild that Yuan Yao is working on in the lab, and it's moving along pretty well. Right now we're at the point where I think we just need quantity of images. So we have deals with a couple of the national primate centers to send us enormous quantities of monkey images. We're going to get those annotated. We're going to get them to annotate each other through a recursive training process. And we're going to basically have the ability to do tracking. I'll just show you this video here, which I think you can probably hear a little bit. We have a deal with the Minnesota Zoo, which has the largest collection of macaques in the United States, 27 macaques in a zoo environment. And these, of course, it's Minnesota, so these are snow monkeys, but they move in basically the same way as rhesus monkeys. They're the same genus, different species. And we put 40 cameras around this cage. These are high-quality telephoto lenses on these cameras that can move around, and they're going to track these monkeys, and we're going to generate enormous quantities of data at the zoo, and then we're going to use that again in this recursive process to generate a system where with only a single camera, you're going to be able to do the tracking and do the lifting. And here's just some preliminary results. Nobody's ever seen this data before. We just got this. I just got this yesterday because I was pestering the Yuan and the other students doing this to get this to me. We can actually do pretty high quality segmentation and annotation with the data we have already. So we're getting there. We're getting to the point where we can do this. And again, we're collaborating with a couple of the primate centers to help them with their science as well with this technology. I won't go into this, but basically we're also, the lifting looks like it's going to work too. This is a pretty well done problem with humans. It's not going to be super duper difficult with monkeys, we don't think either, once we get the basic parts of the model to work. One thing we are doing, if anybody in the audience has enormous databases of other primate species, not macaques, but especially apes are particularly interesting to us. If they can send us those pictures or want to be involved in this, that would be great. Right now we have, only like 10 or 20,000 images. We're looking for like several 100,000. But what we want to do is build a system that can do joint tracking of other species. And what you see here is what the computer does right now. This is state of the art, but we want to make it a lot better. We think we can improve the accuracy quite a bit. Here's our very, very crude first pass video of a monkey, just from a National Geographic video, and trying to track it. So we're not there yet. You can see this is not fully accurate, but this is sort of where we are right now. All right. Oh, look, I'll just do a couple more slides, and then I'll stop talking and I'll answer questions for the rest of the time. What we're going to do with this system, now that it's basically working, now that we can definitely track a monkey in a cage, is we're going to have monkeys move around the lab, and we're not even particularly interested in the type of task. We have a few different foraging tasks, and we're just going to track them, track their behavior, and record brain activity. And so we actually have a bunch of over 100 electrodes using a Charlie Gray drive implanted within the dorsal anterior cingulate cortex while the monkey does this task. The system is not super heavy. It doesn't really seem to affect the monkey's movement that much. The monkey doesn't seem to mind. And we're going to try to relate brain activity in this system to all the motion variables that you would guess that we would talk about based on the previous part of my talk: the position, the velocity, the acceleration, the direction the monkey is looking, all those things. We're going to steal some ideas from Josh Shavitz and Gordon Berman and some other people, where we're going to basically decompose the behavior of the monkey into different behavioral states, and we're going to relate those to each other. We're basically going to do what a primatologist does when they build an ethogram. So a primatologist will sit there out in the rainforest and watch a monkey for months and months and get to know its behaviors, and then they'll start categorizing what the monkey is doing over time in different points of time, and categorizing how long it spends and which one it moves when it moves from one state to another. Except we're going to do that all automatically. So we're going to take the data on the positions of the 13 joints or the 40,000 joints or whatever we have, and we're going to do something akin to UMAP. There's going to be a lot of pre-processing before you get to the clustering stage, but once you do that, then you can just run UMAP on it, and you can detect natural, common, standard behavioral modes, and you can start relating those to each other. So we think of this as basically automating the process of what field primatologists are doing. We can't do that yet with field monkeys. We can only do it with monkeys in the cage because we don't have the ability to track the monkey in the field yet, but we're trying to get there. We have some preliminary data about which states are more likely to transition to other states. I'm not going to walk you through that. I just kind of want to give you an illustration for the direction we're going on this. And oh yeah, here's basically what the monkey's wearing in his head with the electrodes. We're doing data logging, so we basically have an entire Plexon system in the monkey's head. This is pretty standard. A lot of labs are using this nowadays. And that's it. I'm going to stop talking right there because I want to give people a chance to ask questions, and I don't want to go over time. And thank everybody who has attended, and I'll just stick around as long as people want to answer any questions. And if you're feeling shy, send me an email.
Awesome. Thank you so much, Ben. I'm going to take the first question because it's kind of silly, and I'm sure you get asked it a lot. Can you train your monkeys to do the annotation of the poses?
No, you need that. You need much higher accuracy than monkeys.
Seemed like the kind of thing maybe they're good at. It's a natural task, right? Maybe. It's
not crazy. I mean, we have thought about using Turk to do it, but the accuracy even on Turk is not nearly as high as we would need to do it. You just create so many errors, and you need to have other people check for errors, and people to check the people who are checking for errors. Undergrads can't do it. We have professional annotation service that does that, the start of it.
So Rampant Soup asks, you track the tail as well as the limbs. Do you have any plans to extend this to prehensile tails? That sounds harder because it looks like your medial axis model has rigid bones.
It doesn't have rigid bones. It just has relationships that are encoded within it. If the algorithm decides that there's rigid bones in there, that's its own business. We would love to do it with prehensile tails. We just need a data set of like 100,000 New World monkeys that have the right kind of tails. We don't have that. One of the big limitations we're running up against is just the quantity of photos of monkeys. We have, and New World monkeys are just much rarer in captivity than Old World, and you need a huge variety of poses. So you don't just want like 100,000 pictures of spider monkeys or something. You want like 100,000 pictures that are very carefully curated to be as diverse as possible. And we can do that with macaques, and we're working on ways to do that with apes, and you can do that with baboons, and hopefully that will generalize to other monkeys. But the question of prehensile tail is probably we're going to need specialized stimulus sets of spider monkeys or whatever prehensile tail monkeys you want.
Shuafeiyu asks whether based on this model, if there are two videos, excuse me, using the same thing, so I guess two different cameras, trying to track the same object from different random angles, can you infer the viewing angle between the cameras?
Yes, and I didn't go into any detail on that, but that's a very critical part of how our algorithm works. Is it does a lot of triangulation, it does a lot of interpolation, and that's crucial for, basically the way it works is that it makes a guess about where this monkey is in space, and then it uses some other camera to see if that guess is right, and if it's right, then it upweights that connection, and if it's wrong, it downweights that connection, and so it kind of recursively trains itself to guess correctly. And you really need you need the 62 cameras, because for any pose you only have about monkey on about 20 of the cameras at any given time, and so you need about 20 in practice views of the same monkey to make this kind of augmentation work effectively. And that's crucial. We can't figure out a way to do it without that. But once the model exists, then other people are probably going to be able to use it once we release it to the world without the 62 cameras.
So Camille has another question about the tracking, which is how well does it track the multiple monkeys interacting at the same time? And what's the status for tracking social behaviors?
It does quite well. We've never done more than 2 monkeys, but we have lots of tests with 2 monkeys. And it doesn't seem to have any problems. When they start getting cuddling or grooming or anything like that, it doesn't really seem to mess up the algorithm. That's not something we trained in it, but it just seems to work pretty well. So we're pursuing that as well.
Rishi Rajalingam has a question that's near and dear to my heart as well, which is, how do you think about the challenges with respect to covariations between measured variables under naturalistic conditions? Have you been able to reduce the measurements to a set of core or independent or orthogonal variables? Because it seems like a problem for both Pac-Man and the big cage data.
Yeah, that is the problem with, the main problem with moving into naturalistic tasks. And that is, to my mind, the most important reason why we're never going to give up on simple tasks. We're never going to stop being able to do shared tasks, because you always have this multicollinearity problem and correlated variable problem. You basically, we're kind of in a lucky position where we're sort of the first ones doing this, and so we can tailor our questions to the data that we have, at least for the next couple of years. Honestly, probably the next 5 to 10 years, because there's so much low-hanging fruit here. But that is a problem that we have to deal with, and that we're going to have to deal with the Pac-Man task. We've already run up against it, but it does structure the kind of questions you can ask. So that, I will say, very candidly, at the start of my talk, I said you can ask new questions that you can never ask before with naturalistic tasks, but it also does tend to close the door to other types of questions you can ask. And I would say that both types of questions, the types you can answer and can't answer, are interesting. There are ways to solve these problems. You can solve them with huge amounts of data. But right now, we're not dealing with that yet, because we're just trying to solve the technical problems of collecting this data. But I don't want to minimize the seriousness of this problem. This is a very serious problem that we think about all the time.
Lawrence Strickland asks about the generalizability of your OpenMonkey Studio, in terms of whether you can clarify whether the videos of the monkeys in the green room are being used to track the wild or zoo monkeys, or you need like additional annotation.
You need both. So the monkeys in the green room basically gives you the core model, and then you got to augment the model. Basically, If you think about it, deep learning is really, really, really terrible at generalization. It's really awful at it. And so anything you change, if you change even one slight thing, you got to add some new training data to the system. And so going from the green room to the natural environment is going to require a lot more annotated data in the natural environment to generalize it. But on the other hand, what we have, the internal model of the monkey that we got from the green room data does actually a lot of the work, and it greatly, by several orders of magnitude, reduces the difficulty of the problem of tracking in the wild or in the zoo or whatever.
Neurocurious asks whether you have plans to study the changes to foraging behaviors following trauma in the Cayos Santiago colony. And do you think you could capture aspects of behavior that we've missed by doing that?
We don't have plans to study trauma per se, but there are a lot of things we could study, a lot of things we could do. That's not first on our list, but I absolutely do think that this technique of tracking the position of all the joints and then building an embedding structure in comparing those across different conditions is going to be very productive and very fertile, and it's going to tell us things that we couldn't tell using field primatological methods. So I think there's a huge amount of extra information available there, and it's going to let us do very subtle things. Even though the trauma question is one we can't really answer easily because we don't have pre-data.
So Blair Shevlin asks about the Pac-Man task, and whether you can talk more about how you think about decision-making in that context. So with discrete decisions, the decision-making process can be modeled using state space models, like drift diffusion, but it's not clear what sort of process is being used to drive the choice there.
Yeah, that's a good question. Yeah, I'm going to punt on that one, and I'm going to talk to Blair offline.
Okay, fair enough.
It's a good question. It's not a bad question. It's one that merits a longer answer.
Yeah, fair enough. Andy Alexander asks whether he missed whether animals were head fixed during Pac-Man, which I believe you said they weren't, and whether you tried to give the self object on the screen a directional axis, so you could look at egocentric coding relative to the avatar rather than relative to the animal.
We've done it both ways, both head fixed and not head fixed, and the results seem to be pretty much the same both ways. And the monkey doesn't actually wind up moving his head very much anyway, even if his head is free. So that's the answer to that half of the question. The second half of the question is an axis so that we can distinguish egocentric. What is the question again? What was the second half of the question?
Giving the object a directional axis so that you can look at whether the coding is relative to the avatar on the screen or relative to the animal looking at the screen.
Yeah, I mean, I understand the question, but we haven't done that. The monkey in that task is sitting in a chair, even if his head is free to move around, and it doesn't wind up moving around that much because the computer screen's not that big. It's not that much benefit in moving your whole head. Mm-hmm. And if you do move your head, you don't move it very much. And so we can't really disambiguate these different types of things. We just don't have the analytical leverage to do it because there's not enough variability in the independent variables. So those are really good questions. And I would say these are the reasons why Pac-Man is not enough. We want to make it even more naturalistic and do big cage kind of stuff. Even if that, we introduce a lot more collinearities and things, as Rishi's pointed out. Yeah, so it's a good question. We don't have the answer to that.
Neurocurious asks, related to their previous question, which was about the trauma and the foraging task, whether historical video can be analyzed using the Open Monkey Studio.
Yeah, I think it could be. I think it could. Yeah, I mean, I would want to see exactly what the video looks like, but that's actually not such a terrible idea. The question is just how much quantity and quality of data there is in the video record. So, it would depend on the specific circumstances. But in theory, if there's enough data, it should be doable.
And then also about the network, I have a question about, have you thought about modularizing the model, excuse me, so that you could segment out the monkey in the studio and the zoo with different networks, and then ask a third network to give you the medial axes of the joints. That way you need fewer samples to train the segmentation only model on.
I don't know the answer to that. We'd have to talk to Yuan about that.
Okay, if there's any other questions, we can ask them. Otherwise, oh, wait a second. Otherwise, I want to thank Ben again. Someone says that they wish they'd seen this talk before. I would have been more ambitious in my postdoc project. I feel like that's always the way when you see new work. You're like, why didn't I do this? Yeah, I want to thank Ben a lot for giving this talk. I've always been really inspired by the work he's doing. I think he's doing a lot of really great stuff. Every new paper I'm kind of inspired by. So thank you again for talking. And thank everyone for the questions.
Yeah, thanks for the invitation, and thanks everybody for coming. And I hope to hear from several of you soon.